this may not be applicable for a lot of the states, but obviously I think most people know in Texas we have a very active captive deer breeding community out there, and so the Holy Grail for trying to manage CWD and combat CWD is to find that those tissues and the amplification techniques or whatever technique to detect the disease or the prions with a high degree of specificity and sensitivity as early as possible. So thanks to you, Dr. Morales and Dr. Nichols, for providing those samples and continuing to pursue those techniques. So hopefully we can eventually find something that will get us there much quicker, less expense, obviously, and tissues or samples that are easy to obtain. So we'll keep our fingers crossed, and Texas is going to continue to do what we can to help provide those samples. We wish we didn't have any CWD positive samples, obviously, to contribute to the cause, but we definitely support the work. Our next presenter, the last one of this session, is Dr. Michael Samuel. He's a emeritus professor in the Department of Forest and Wildlife Ecology at the University of Wisconsin in Madison. He served as a wildlife research biologist with the U.S. Geological Survey for 30 years and professor of wildlife ecology at the University of Wisconsin-Madison. He was assistant unit leader at the Wisconsin Cooperative Wildlife Research Unit from 2003 to 2017. Prior to that, he worked on wildlife disease problems for 15 years at the USGS National Wildlife Health Center. He has a BS degree in computer sciences from UC Berkeley and an MS in ecology from UC Davis and an MS in applied statistics and a PhD in wildlife from the University of Idaho. His research interests are primarily in wildlife diseases with focus on disease ecology and management, epidemiology, disease transmission and modeling, and wildlife population impacts. He has conducted research on chronic wasting disease in white-tailed deer in the Midwest since 2002, avian malaria and pox in Hawaii forest birds, avian cholera, botulism, influenza, and lead poisoning in waterfowl, as well as sylvatic plague and vector-borne diseases of wolves and mesocarnivores in Wisconsin. He has served as assistant editor for the Journal of Wildlife Diseases for the past 15 years. Please help me welcome Dr. Samuel.
The last time I was in this part of Texas was in 2009 for a spring turkey hunt up near Mason, and happened to get a tick bite, and later came down with a viral encephalitis that sort of put a crimp in my style for about 6 to 9 months. So I was kind of reluctant to come back to this part of Texas, but Brian Richards sort of twisted my arm and said, well, it's winter, and we're going to keep you inside anyway, so no risk, and probably I'm immune to whatever it was I got before anyway. So thanks for the opportunity to come and talk about some aspects of chronic wasting disease. I'm going to talk about transmission of the disease, what we know about it, and probably even more maybe allude to what we don't know and need to know about this disease as well. So CWD occurs in lots of deer tissues. What we think about this disease is that it's ingested. Infectious prions are ingested. They get into the gut. They get through the Peyer's patches or other lymph tissues in the gut, and then somehow get distributed around the body, particularly in the nervous system, and then eventually get to the brain where they cause disease. Until they get to the brain and actually advance a bit in the brain, there's really no disease symptoms that the animals are exhibiting. So some of the tissues, the lymph tissues in particular, the retropharyngeal lymph tissues, are detectable. We can detect prions in them fairly early on, within about 3 to 4 months, typically in white-tailed deer. And then the other part is that these tissues and things in the body then shed or secrete prions, which then are the route of transmission to other animals and to the environment. So in particular, the things that the secretions that are infectious are saliva, feces, blood, and urine. So pretty much anything that comes out of a deer is going to be potential for transmitting this disease. So what we know about the rate of accumulation in tissues in the various species is is not the greatest, but I think there are some general patterns that we can see. For example, elk seem to have lower prevalence than deer in wild populations. And I think you've seen that today in talking about some of the populations of elk being maybe around 10% or 15% prevalence, whereas deer can get up to 50% and higher. Elk seem to have lower levels of brain deposition and higher levels of brain deposition and more variability than deer do. And they seem to have lower levels and more variability in the tonsils and in other lymph tissues as well. And less deposition in the retropharyngeal lymph nodes. My sort of understanding of elk, and I'm not certainly don't follow elk that much, but from Mike Miller, is that you want to probably test both the retropharyngeal lymph nodes and the brain to be certain that you're detecting the disease adequately. Our deer species, the mule and white-tailed deer, behave a little differently in that regard. They tend to get higher infection rates, particularly in captivity. We've certainly had infection rates that have been 80% and higher in captive populations. They seem to have a lower level of brain deposition, and they seem to have early and high deposition in their lymph tissues, which makes it handy for detecting infection. So what do we know about the relative infectivity of these different secretions that deer have? In general, I think we know a fair amount from the tests like Dr. Morales was talking about, PMCA and other tests like that. Saliva seems to be the most infectious secretion that deer have in particular. Blood, I think we're a little less certain about. That seems to be infectious as well. Where that falls in the spectrum is still a little bit unclear. And then below that are feces, and below that yet again are urine in terms of what seems to be the amount of infectivity or infectious material that are secreted by those different agents. So how does transmission occur, though? We have these infectious secretions that occur. Well, one way, deer are very social critters. They tend to rub noses, they tend to have mutual grooming, they tend to interact with each other quite a bit. So one of the probably very important ways in which the disease is transmitted is by contact. And that's probably by things like saliva and urine, mutual grooming, certainly likely to deposit saliva and things on other deer, and also potentially blood. And I'll come back to blood in a little bit, or one potential route for blood transmission. In addition, environmental contamination, you've heard that a lot today. Environmental contamination is a potential issue with this disease as well. Infected carcasses that die in the landscape can decompose and then deposit prions on the landscape. And you've heard that at least in the lab, people have been able to show that plants can uptake those prions and store them. Places like feeding and baiting sites where animals congregate, where they're sticking their noses in places, where they are basically depositing saliva or defecating. Those can then present sources of environmental contamination as well as mineral licks, which seem to sort of follow the same kind of pattern. And of course, some of these things are why when we have areas with CWD that feeding and baiting is banned or at least strongly discouraged. Scrapes. So the scrapes that deer make during the breeding season, they basically rub areas on the ground and urinate in them and defecate in them to sort of make other bucks know about their presence are a potential route. I think not anybody has shown that yet, but that's a potential route. As well as food sources, general water, although there's a big dilution effect there potentially. And deer also consume quite a bit of soil. Not only at mineral licks, but in general as part of their diet. So that's another potential route. And as Dr. Morales pointed out, plants, I think, are something we need to be worried about as well. Unfortunately, at this point, we don't know the magnitude of these two routes of transmission, the direct route and the environmental route, and how those might change over the course of an epidemic. And I think that has a lot to do with what we think about for how to manage this disease early and late stages. So the reason we sort of identified that direct and indirect or direct and environmental contamination are important routes of transmission was from a captive study that Mike Miller and company did in Colorado. Now, these are done in highly contaminated environment, but basically what they showed is that if you put naive deer with infected deer over here, about 20% of them become infected just by this contact sort of process. If you put them in with infected carcasses, that about a quarter of them become infected. And if you put them in with just residual excreta, urine, feces, and things that contaminate the pen, about 10% become infected. So we know that these routes work in a captive situation. What we know much less about is how they work in the wild situation. So another important part of this, we've talked about, okay, these are the urine, feces, and saliva are three major routes of infection, potential infection. So when are animals shedding those detectable levels of prions? So for white-tailed deer, we know more probably about white-tailed deer than the other species at this point. But we know that prions are being shed in urine as early as 3 months post-infection. In feces as early as 6 months post-infection, and in saliva as early as 3 months post-infection. So way before these animals are symptomatic, or we can tell that they're infected, they're shedding prions, both then potentially with contacting other animals and into the environment. We know less about mule deer at this point, and these represent what people have documented as earliest detection. It doesn't mean that these mule deer aren't excreting infectious prions earlier as well, is that people haven't documented it yet, and maybe it's partly because they haven't tested it rigorously as well. But mule deer seem to be, at this point, later in terms of shedding in urine and feces, and we don't really know much about saliva in either urine or elk at this point. And then elk have been shown to shed in feces very early post-infection as well. So there's lots of potential for transmitting this disease via these routes very early in the process. Okay, so I mentioned blood. Well, deer don't get transfusions, right? They don't go into the hospital. But it has been, people have reported, some of my biologist friends have reported to me that male deer tend to lick the bloody pedicels of other deer as they've shed their antlers. I suspect this could happen a little more in captivity, and maybe some of you who have raised deer maybe see this maybe perhaps more than I would see it in the wild. We don't really know how common it is, but the potential for uptake of blood from an infected animal would be there. So what do we know about susceptibility of these animals? So deer appear to be more likely to be infected than sympatric elk, and again, that's exhibited through the prevalence patterns that we've seen, higher prevalence in the deer species than we see in elk. The white-tailed deer seem to have higher rates of infection than mule deer. I think that might be still a bit debatable, depending on what system people are working in and what their experiences are. And then the other thing that's really important here is these genotypes that Dr. Morales mentioned. So we know that the analogy is for people, what drives blonde hair versus brown hair, what drives blue eyes versus brown eyes, what drives baldness and gray hair, I don't know, but that's maybe somehow in there as well. But there are different genotypes that have different levels of susceptibility to the prions. But I think it's very important at this point in time, we don't know of any genotypes that are actually immune to the disease. They all get infected. It's just a matter of how long it takes to get infected and what happens to them post-infection. So this is a bit of a hypothetical, but probably not too far off graph of 3 different genotypes in white-tailed deer, which Dr. Morales started to talk about as well. So the 96GG is probably the most susceptible genotype that we know about in white-tailed deer. The 96GS is probably intermediate, and the SS are probably the most resistant to infection. So what we think happens is that basically these animals become infected at different rates, and then the course of the disease for them seems to be different as well. the more resistant animals seem to live longer after they become infected as well. And that raises lots of concerns about transmission, because are we potentially having these more resistant animals out on the landscape, depositing more in the environment, potentially transmitting the disease to other animals as well? We don't really know the answer to that yet. We need a lot more information about it. We published a paper last year only suggesting that perhaps these more resistant genotypes might not be shedding quite as much as the more susceptible genotypes. So this is definitely an area where we need to know a lot more, but this has lots of implications for where we're going in the future with this disease. So why is this route of transmission important? Well, environmental reservoirs are long-term. We know that the prions tend to last in the environment. They're nearly indestructible. They last in the environment for a long time. So setting up long-term sources or reservoirs in the environment is a challenge for the long-term management of this disease. For the short-term management, then we have to also decide what's the best strategy. Do we need to control the animals, or do we need to somehow do something about the environment? What should we be doing to help manage this disease? And I think at this point, as I said, we have a ways to go in terms of learning about that. So what does the science tell us at this point about some of the aspects of this disease in terms of the environmental sources? So Dr. Nichols published a paper a while back demonstrating that prions could be found in water runoff areas in Colorado. We published a paper this year where we found prions in mineral licks in our endemic area in Wisconsin. The majority of the mineral licks that we sampled had detectable levels of prions in them. And as you can see from here, this is kind of a mineral lick. They could be natural mineral licks or artificial mineral licks, artificial ones created, let's say, for cattle, salt and mineral blocks, things like that, that deer will consistently use. They'll drink the water in it, they'll eat the soil, they're salivating in it, they're urinating in it, they're defecating in it. Other deer are then coming in, so deer, we think that what's going on is that deer are depositing prions in those highly used sites, and then they're there for other deer to consume. And in addition to, Dr. Morales talked about some of the issues related to inhibitors in blood. There are severe inhibitors in detecting prions in the soil as well. Things like organic matter and things like that make it very difficult for us to actually do environmental testing at this point until we can learn a lot more about that. So uptake by plants, this is a huge concern, obviously. Lab experiments show that there's kind of mixed lab experiments on this, I have to say. They're all from the lab at this point, showing that early on that they could be that wheat could attract and attach prions to the roots, but not to the stems. And then other research later on showed that, in fact, it can be uptake into the stems as well, and also adhere to the plants. If it's deposited on the outsides of the plants, it can actually then adhere pretty rigorously to the outside. And this is an important and pretty much unknown, I think, potential route of transmission. And as Brian Richards pointed out, it certainly got the attention of Norway and people in Europe in terms of agricultural products. Not that we have the science to demonstrate that any of this is happening in the field yet, but as a precautionary principle, then they're saying, well, we're going to either ban or restrict agricultural products potentially from areas that might have this disease. So I think this is a potential big game changer for a lot of people and the agricultural people to think about as well with chronic wasting disease. In terms of longevity in the environment, we don't know a lot about CWD, but we know more about scrapie. Scrapie disease was as a prion disease in sheep. Scrapie prions that were buried in the soil, buried in the ground, lasted for 3 years, at least 3 years. And it's been reported that a sheep ranch or sheep farm with scrapie that was depopulated and sheep were allowed back on after 16 years became infected as well. So we think that prions last very long time. And so these environmental sources of contamination are really important. And also then, we know that it lasts at least 2 years from a study that Mike Miller did. So this is sort of my hypothetical model based on sort of what I've seen in Wisconsin and what we sort of have learned from the science in Wisconsin about what might be happening with these two routes of transmission. And I'll talk about some of the evidence for that. But what I think is happening is that early on in a disease cycle, and notice first of all that these disease cycles for CWD are long. They're not going to be here and done within 10 years. They're here to stay. And it's a long term for those of you involved in management, it's going to go well beyond your careers. But I think that basically early on in the disease cycle, that direct transmission between animals is probably the driver of infection. And then later on in the process, and after we've accumulated a number of infected animals on the landscape, and we've had an opportunity to deposit infectious prions in the landscape, then environmental contamination is likely to pick up. And then those two become additive and we get higher rates of transmission occurring because of that. And whether the environment actually overtakes direct transmission at some point, that's just, I don't know, maybe direct remains larger, but certainly environmental contamination can be a big issue potentially for the future management of this disease. So what are the evidence that we have for these various routes of transmission? Well, there was a study published from Colorado which showed that clay in the soils, now we know that prions bind to clay, and we also know that when they bind to clay, they make the prions more infectious to animals. We don't know that for CWD, but we know that for other prions. So the Colorado study, I'm one slide ahead of myself here. So the Colorado study that Miller did showed that basically direct and indirect worked. And then the study by Dave Walter et al. Showed that in areas in Colorado that had higher clay content, they also had higher rates of CWD infection. So they basically showed at least a correlation or a somewhat more of a link between the soil characteristics and infection of chronic wasting disease. The evidence for this is mixed. There's a study in Illinois published a couple years back indicating that basically soils with less clay and higher sand had higher prevalence of chronic wasting disease. And we did 2 investigations at different spatial scales in Wisconsin trying to look for soil characteristics, particularly clay, as being related to infection, and couldn't find any evidence. So I think the evidence for whether environmental transmission is going on and how much is not very strong at this point. Okay, what about evidence for direct transmission? So we published a study quite a while ago now in Wisconsin where we showed, using genetics, that female deer are more likely to get disease from their maternal social group than they are from just being in the same environment where CWD occurs on the landscape. So there seems to be a connection between who you relate to, who you interact with, and your likelihood of getting chronic wasting disease in Wisconsin. We've also recently published a study where we showed that males have 3 to 4 times higher rate of infection than females, but we know that males don't have 3 to 4 times higher home range, larger home range in Wisconsin. They have about a 1.3 to 1.5 larger home range. So if environmental transmission, just being out in the random environment, was driving transmission, we wouldn't expect transmission to be as high as it is in males. In addition, we've also shown that CWD is a frequency-dependent disease rather than a density-dependent disease, and that's a whole other talk unto itself. But frequency-dependent diseases really tend to occur in things that have social limitations. So like human sexual diseases, for example, have limitations. We call them frequency-dependent diseases because there's only so many interactions you're going to have. You're not going to interact with everybody in the population. So that's another suggestion that direct transmission is important. One of the caveats to this, 5 minutes, okay? One of the caveats to this is fawns seem to have a lower rate of infection or lower prevalence than their mothers. So why is that? That's one of the biggest pair bonds we have, right, in deer, is between a mother and its fawn. So I don't have a good answer for that. It may be related to susceptibility a bit of young animals, or it may be that it's partly related to environmental exposure, which would be higher in females. So what do we need to do? Some of the things we need to do is we need, Dr. Morales talked about some of these. There are methods for improving our detection of prions. We have lots of challenges in terms of extracting prions from different environmental matrices, from feces, from blood, from soil, from plants. These are all challenges that are ahead of the people working on these very sensitive methods. One of my concerns is that it's easier, in my book, to look at environmental contamination and detect environmental contamination than it is to look at direct transmission. Looking at direct transmission in a disease that you don't really see till 2 years later is enormously difficult. And so I want to make sure that we're keeping that in mind, and that when we talk about environmental contamination, we're not jumping to the conclusion that that's the major route of transmission, because it may not be. So some management and research needs: we need to know better about these relative importance of direct and indirect transmission, because it affects our long-term management. We need to improve how if we can, figure out ways to measure direct transmission by animal contact and proximity. We need to work on these environmental reservoirs and how they provide transmission to our deer species and potentially to other animals as well. There's other animals that use these mineral licks. There's other animals that use feeding areas as well. Any of those animals that might be susceptible to prion diseases could be at risk as well. We know not enough at this point about infectivity, particularly in the different genotypes and the timing of excretions and secretions, and shedding in those animals as well. And then in general, CWD detection methods. So the kind that Dr. Morales talked about would be great to have more of. So I guess we're going to hold questions till later. Thanks.